Hey guys, Assalamu alaikum. I hope you all are doing well. In the last video, in part one of bioenergetics, we had started with we had done till glycolysis and we had done till the net gain from glycolysis. Okay, so let me just revise the net gain from glycolysis for a bit. Okay, so basically the net ATP which are produced by glycolysis are two. Okay, why? Because the total ATP produced are four, but two of them are used in the energy investment phase, as you can see over here, right? In the preparatory or the energy investment phase, in the first step we see that this ATP is converted into ADP, where glucose is being converted into glucose 6 phosphate, right? And another ATP is converted into ADP when fructose 6 phosphate is converted into fructose 1,6 biphosphate. Okay, so these are the two ATP which are used, right? Now, how many ATPs are produced? We are going to see this right now. Okay. After that, we studied about isomerization and how G3P and DHAP are being interconverted into one another. That is why we now have two molecules, right? Two molecules means two ATP are produced at every step. Okay. When 1,3 bisphosphoglycerate is converted to 3-phosphoglycerate, it removes a phosphate, and this phosphate is added to ADP in order to form what? It forms ATP, right? Similarly over here, so hence at step number 7, how many ATP are produced? 2 ATP are being produced. And in the last step, where PEP, phosphoenol pyruvate, is being converted into pyruvate with the production of ATP, how many ATPs are produced? 2 ATP are produced, right? So when 2 and 2 ATPs are produced, the total are 4 ATP, and 2 of them are used already, right, in the preparatory phase. So we're going to minus 2 ATP. That produces a net yield of 2 ATP. In addition to this, we have got two molecules of pyruvate from glycolysis, and over here, as you can see, two molecules of water, and we have two molecules of NADH. See, on every side we have got two molecules of NADH, right? So this is the net yield of glycolysis. I hope you all understood that. If not, please let me know in the comment section. We're gonna do it again together. All right. Next, we move on to the link reaction. Now, what happens after glycolysis? We have a product produced that is known as pyruvate, right? Now, after glycolysis, which thing occurs? Krebs cycle occurs. Krebs cycle occurs, but it needs a substrate that is known as the acetyl-CoA. Right now, we have got pyruvate, but it needs acetyl-CoA. So, what are we going to do? We are going to convert this pyruvate into acetyl-CoA with the help of a reaction that is known as the link reaction. Now, this link reaction occurs in the mitochondria, in the mitochondrial matrix, in eukaryotes, right? And it is catalyzed by the pyruvate dehydrogenase complex. Pyruvate dehydrogenase. Okay, so what does this mean is that this is basically oxidative decarboxylation. Now, why is it given this name? Because you see, pyruvate over here has got 3 carbons, but acetyl-CoA has got 2 carbons. So there is a removal of carbon. There is decarboxylation because carbon dioxide gas is being produced over here, and also it is oxidative because NADH is being produced, right, from NAD positive. That is why it is given the name of the link reaction between glycolysis and Krebs cycle, and hence oxidative decarboxylation, right? It produces, the yield of this is that it produces one molecule of carbon dioxide, acetyl-CoA, which is later used in the Krebs cycle, and it produces NADH, right? And it uses coenzyme A in order to form acetyl-CoA, right? Now let us see all the steps. The first step is basically decarboxylation. What happens over here, as I just explained to you, that this 3 carbon molecule pyruvate, it loses 1 carbon as carbon dioxide, and now a 2 carbon fragment remains only, and this is oxidized where NAD positive, wait, let me just, yeah, where NAD positive is being converted into what? It converts into NADH. Okay. The second step is the formation of acetyl-CoA. So now this 3 carbon fragment of pyruvate has converted into 2 carbon fragment, right? Now this 2 carbon fragment, it combines with coenzyme A in order to form acetyl-CoA, right? In chemistry, you have studied that 2 carbon, two carbon substance is given the suffix of acetyl, right, or ethyl or acetyl, right? So when this ethyl or acetyl combines with coenzyme A, it forms acetyl CoA, right? This is the whole overall reaction. Okay, pyruvate plus NAD positive plus coenzyme A, it forms NADH plus carbon dioxide and acetyl CoA. This will be used for the Krebs cycle, which we are gonna study right now. Okay, I hope this is clear. If you have any queries, let me know. All right. Now let's keep our diagram over here and I'm going to explain it to you together. Now Krebs cycle is a cyclic series of enzyme catalyzed reactions in the mitochondrial matrix. So that reaction, this reaction, the link reaction where pyruvate is being converted into acetyl CoA, it occurs in the mitochondrial matrix as well. And this Krebs cycle also occurs in the mitochondria. Okay, now acetyl CoA is oxidized to carbon dioxide with the release of these molecules NADH, FADH2 and GTP or ATP that drive ATP production in oxidative phosphorylation. So what happens over here, there are basically energy molecules which are being produced by the action of the Krebs cycle. Okay, acetyl CoA is a two carbon compound that combines with three molecules of NAD positive, FAD, GDP, phosphate inorganic, and 2 water. In order to form two molecules of carbon dioxide, this NAD is converted into NADH. FAD is converted into FADH2 and GDP it is converted into GTP. Guanosine diphosphate converts into guanosine triphosphate. Why? Because we have a phosphate inorganic molecule over here which it takes in order to convert into GTP plus this coenzyme. Okay. So let us move on to the steps. Let us take it step by step. Okay, so the first step is condensation. The substrates are acetyl CoA and oxaloacetate. Okay. After that, what happens? The enzyme is citrate synthase, and the first step leads to the formation of citrate, which is irreversible and it is a key regulatory step. So we are gonna start from here. This pyruvate is converting into acetyl CoA. This was basically the link reaction. Okay, and after that this acetyl CoA is converted into citrate with the help of which molecule? With the help of oxaloacetate. Over here oxaloacetate is missing. Oxaloacetate, acetyl CoA plus oxaloacetate with the help of the enzyme citrate synthase. You don't need to remember the enzyme, it's not important for your level. I'm just telling you so that you gain more knowledge, you know. So then it produces citrate, which is a 6 carbon compound, okay? And it releases this coenzyme A, right? And it requires water for this step. The next step is isomerization. As you see over here, isomerization. What happens is that this citrate converts into isocitrate, okay? When the position of the carbon molecule moves, when the position of the bonds move, what happens is that isomers are produced. Why? Because they have the same chemical formula, but they have got different structural formula, right? So citrate produces isocitrate, which is a 6 carbon compound, and the enzyme used over here is the aconitase, okay? This prepares the molecule for oxidation by exposing a reactive hydroxyl group. So what happens is that the position of the hydroxyl group changes in such a way that it becomes reactive. Okay. So when it would react, it would produce more reactions. Okay. After that, what happens for the third step? The third step is known as the first oxidative decarboxylation. Why is it known as that? Oxidative, because this NAD positive is converted into NADH, right? That is why it is given the name oxidative. Decarboxylation, why? Because carbon dioxide is removed, right? It means that one carbon molecule is being removed. That is why decarboxylation. ठीक है, you have to learn it by its name. First, oxidative decarboxylation occurs when isocitrate is converted into alpha ketoglutarate. Alpha ketoglutarate is a 5 carbon compound. And why is it a 5 carbon compound? Because one molecule of carbon is removed as carbon dioxide. Okay, sorry, no, one atom of carbon is removed as carbon dioxide, right? After that, This is basically the rate limiting step of the Krebs cycle. Highly, highly important for your MCQs. Right? That where does first oxidative decarboxylation occur? What is the substrate for that? You're gonna say that it is isocitrate. What is the product for that? It is alpha ketoglutarate, which is a 5 carbon compound, right? And isocitrate was a 6 carbon compound. Okay, and then what happens over here? NADH is being produced. One molecule of NADH is being produced, and this is the rate limiting step of the Krebs cycle. The second, the step number 4 is second oxidative decarboxylation. Now, this occurs when what happens? Basically, alpha ketoglutarate, it is converted into succinate. Okay, this is a 4 carbon compound. Why? Because this is second oxidative decarboxylation, which means that now I have got 5 carbons. From 5 carbons, 1 carbon will remove, and then it will be converted into a 4 carbon compound that is known as succinate. Okay, see, carbon dioxide is being removed over here, as you see over here, right? And one NAD positive is converted into NADH. This is second oxidative decarboxylation. And also in this step, what happens? Succinate is produced, right? First, what happens first over here? Succinyl CoA is being produced, which is missing in this, cycle. But after that, we're gonna see that this succinyl CoA is being converted into succinate, and in this step what happens? The GDP is being converted into GTP, guanosine diphosphate plus 1 phosphate inorganic is being converted into guanosine triphosphate, right? And ADP is being converted into ATP. Okay, so this is how the phosphorylation occurs, as we read over here, right? First, what happened? We had alpha ketoglutarate, which was what? 5 carbon, right? Then what happened? Then we had a second oxidative decarboxylation in which one NADH is being produced, and since it is decarboxylation, so carbon dioxide is being removed as well, and we produce what? Succinyl CoA, which is what? Which is a 4 carbon compound. Now the succinyl CoA, it will remove this coenzyme A, and it forms what? It forms succinate, which is also a 4 carbon compound. But what happens over here when succinate is being produced? It produces one GTP in animals or one ATP in plants and bacteria, right? So if we had GDP, it is converted into GTP, right? Because we have one phosphate inorganic that either adds to this GTP or it either adds to ADP, and it converts it into ATPs, right? So in humans or in animals, GTP is formed, okay? Whereas in plants, ATP is formed. So this is a highly important point for your MCQ point of view. The only step that directly generates a high energy phosphate, which is known as substrate level phosphorylation. This process, this step is known as substrate level phosphorylation. Let me just clear everything over here so that I can, we can revise the cycle. Okay. So this is. First, we move on to oxaloacetate plus acetyl-CoA. It produces citrate, right? Now, this is first isomerization. Citrate is converted into isocitrate. Isocitrate converts into alpha ketoglutarate. This is first oxidative decarboxylation, and this is also the rate limiting step. Okay. Secondly, alpha ketoglutarate converts into succinyl CoA, which is second oxidative decarboxylation. Succinyl CoA converts into succinate. This is the process that is known as substrate level phosphorylation. Why? Because at the level of the substrate, at the level of ADP or GTP, phosphate is being added. Phosphorylation happens in order to produce an energy compound that is known as the GTP or ATP. Right? After production of succinate, what happens? It is being converted into fumarate. Now, fumarate is also a 4 carbon compound, right? The enzyme used over here is known as succinate dehydrogenase, which is also a part of the electron transport chain, complex number 2. And what is produced during this process? FADH2 is being produced. FAD is converted into FADH2. Okay, you can remember it like that, that when fumarate is produced, fumarate starts with F, so FADH2 is also produced, which is also starts with F. Okay, so this is a common similarity between them. This is a membrane bound enzyme and it is directly linked to the electron transport chain. Okay, so from over here, the succinate dehydrogenase, it is directly linked to the ETC, electron transport chain, right? And now what happens over here, fumarate gets hydrated. One molecule of water is being added to fumarate and it produces malate. Malate is also a 4 carbon compound, right? So now you see that the number of carbons are not changing after succinyl CoA. They were only changing during oxidative decarboxylation. Okay, first they were 6, then it became 5, then it became 4. Okay, after that it would stay constant throughout the rest of the cycle. Okay, so now what happens over here is that this malate, water is added to fumarate in order to produce malate, which is a 4 carbon compound, and the enzyme is fumarase. Okay, it prepares the molecule for final oxidation. Now, after malate is being produced, what happens is that this malate is converted into oxaloacetate. Okay, with the enzyme malate dehydrogenase. Dehydrogenase means that malate would remove its hydrogen, dehydrogenate itself, remove its hydrogen, and this hydrogen is added to NADH. Okay, so what happens over here is that one NADH is also being produced. After that, oxaloacetate is produced, and oxaloacetate again combines with this acetyl CoA to produce citrate, isocitrate, alpha ketoglutarate, succinyl CoA, succinate, fumarate, malate, oxaloacetate. So we completed the whole Krebs cycle. Okay, now we are gonna do the net yield of energy per acetyl CoA. Wait, let me just erase this so that we can do this. All right, so let's move to the net yield. See, net in the net yield we have some molecules. For example, see, the first one is NADH. Okay, one, two, three. Three NADH are being produced. Okay, so how MCQ comes in your MDCAT? Basically, they're gonna ask you where NADH is being produced. When isocitrate is converted into alpha ketoglutarate, when alpha ketoglutarate is converted into succinyl CoA. These two steps are where NADH are produced. And the third one is when malate is converted into oxaloacetate. So let me write over here. NADH, it is produced when what happens? First is isocitrate converting into alpha ketoglutarate. Right? Second one is when alpha ketoglutarate converts into succinyl CoA. And the third one is when malate is converted into oxaloacetate. They're going to ask you where FADH are produced. So since we remember that F means fumarate, so it means that when malate is converted into fumarate, right? Where is GTP produced? GTP is produced where succinyl CoA is converted into succinate. This is known as what? This is known as substrate level phosphorylation. As you can see in the diagram, this is known as substrate level phosphorylation. Where is carbon dioxide being produced? It is produced at the level of oxidative decarboxylation. First, oxidative decarboxylation occurs at the level of this isocitrate converting into alpha ketoglutarate. This is first. And the second one occurs when alpha ketoglutarate is converted into succinyl CoA. So these are the steps where carbon dioxide is being produced. I hope this is clear. We are done with the Krebs cycle. Let's move on to the significance. Let's just read the significance so that it is clear. Okay. So the significance of the Krebs cycle is that it is a source of reducing equivalents. We have got lots of reducing equivalents, NADH, FADH2, that basically drives the electron transport chain and it helps to produce ATP. Okay, so what is the use of Krebs cycle? Just mainly to produce energy. And it provides intermediates for biosynthesis. Okay, it provides intermediates that produce amino acids, porphyrins, fatty acids, not that important. It is a central hub of metabolism, carbohydrates, carbohydrates, fats, proteins, all of them, they produce acetyl CoA. Okay, all of them are converted into C. First, what happens? Glycolysis occurs. During glycolysis, glucose, basically the main energy molecule, converts into pyruvate, right? And after the Krebs cycle, when we need them from Krebs cycle, this pyruvate is converted into acetyl CoA. Okay, so all of them, they're saying that all the compounds of energy which we eat, they are converted into a common compound that is known as acetyl CoA. And after acetyl CoA, these are further being metabolized. Okay, now let's move on to the electron transport chain. All right. So now what is electron transport chain? It is a series of protein complexes and electron carriers that are located in the inner mitochondrial membrane. It is very important to learn the location where these steps are occurring. You need to remember the location of glycolysis, location of the conversion of pyruvate to acetyl CoA, that is the linking reaction, location of the Krebs cycle, and now the location of the electron transport chain, which occurs where it occurs in the inner mitochondrial matrix, and it transfers electrons from NADH and FADH to oxygen. Okay, that helps to produce a proton gradient. So basically, it is a chain of electrons where electrons are transferred from one complex to another complex, and it drives what? It helps to produce ATP at the end, right? So you see over here that this is the outer membrane of the mitochondria, this is the intermembrane space, and over here we have the inner membrane. Now what happens? This reaction occurs in the inner membrane. Why? Because all of these complexes, complex 1, which is NADH and coenzyme oxidoreductase complex, okay, complex 2 is the succinate dehydrogenase, which we studied earlier when we were studying about succinate, succinyl CoA being converted into succinate. So this is the complex succinate dehydrogenase, which is directly used in the electron transport chain, this one, and it contains coenzyme Q. Complex number 3. Okay, so this coenzyme Q is also known as ubiquinone. Please remember the name. Complex 3 is cytochrome 3bc1. Okay, you need to remember this. Complex 4 is cytochrome C oxidase. Okay, so complex 3 is cytochrome BC. Complex 4 is cytochrome C oxidase. Okay. After that, we have got ATP synthase, which basically drives the whole proton gradient and it produces ATP. Okay. Alright, so now what happens in complex 1? It basically accepts two electrons from NADH and transfers them to coenzyme Q. So this NADH, it is being converted into NAD plus two hydrogens, and it produces two electrons. Okay, two electrons produced here are transferred to coenzyme Q, which is known as ubiquinone, and it pumps four hydrogen into the intermembrane space. Okay, so four hydrogen are pumped down and two electrons are transferred from the first complex, which is NADH dehydrogenase. Okay, NADH basically gives out its hydrogen. Okay, it gives out its electrons, and these electrons are transferred to ubiquinone, which is coenzyme Q. Okay, now what is complex 2 doing? Complex 2 is doing what? It basically takes FADH. Okay, complex 1 was for NADH, complex 2 is now for FADH. So now FADH2, it also gives in hydrogen ions. It also, takes in, it also converts into FAD plus 2 hydrogen. Okay, and it gives 2 electrons, where it gives 2 electrons to coenzyme Q. Protons are not no protons are pumped over here. This is the same enzyme as in Krebs cycle. Okay, wait, where succinate was being converted into fumarate, what happened? We had FADH2 being produced, right? Now this FADH2, it is converted into FAD plus 2 hydrogen positive, right? And also it gives 2 electrons, where it gives 2 electrons to coenzyme Q, right? So now this coenzyme Q has got 2 electrons from complex 1. It got 2 electrons from basically the conversion of NADH to NAD positive, NAD plus 2 electrons, right? And the same way it had got 2 electrons from FADH2, which also produced two of them, right? So it has a total of four electrons. It collected these from complex 1 and complex 2. The first complex name was NADH coenzyme Q oxidoreductase complex, as written over here. And complex 2 is directly linked to the Krebs cycle, and it is known as succinate dehydrogenase complex. Okay, so now this ubiquinone is a lipid carrier. It is a mobile lipid carrier. Mobile means it can travel from one place to another. Why is it lipid? Because it is present in the membrane. Okay, it is present in the mitochondrial membrane. Now you have now you know that membranes are made up of lipids, right? So this is also made up of lipids so that it can easily dissolve and it can easily travel from one point to another. It collects electrons from coenzyme 1 and 2 and it delivers to complex 3. Okay, so now it took 4 electrons from complex 1 and 2 and it delivers these electrons to complex 3. Complex 3 is known as cytochrome BC1 complex and it accepts electrons from coenzyme Q and these are transferred to cytochrome C, which is a small mobile protein. Okay, so now this is made up of protein and this helps to basically complex 3 is a relay structure that takes electrons from coenzyme Q and it transfers them to cytochrome C and it pumps 4 hydrogen into the intermembrane space. You need to remember that it is pumping in 4 hydrogen, right? Now what is cytochrome C doing? It is small and it is soluble in proteins and it shuttles electrons one at a time from complex 3 to complex 4. It basically transfers electrons where from complex 3 to complex 4, as you can see over here, that two hydrogen plus oxygen, they get added with two electrons in order to produce water molecules. So what is being produced over here? Water molecules are produced at cytochrome C. Okay, they are transferred to oxygen. As I just told you, oxygen plus 4 electron plus 4 hydrogen positive produces 2H2O. Okay, so the reaction over here in this picture is written differently. Wait, it is written 2 hydrogen positive plus half oxygen plus 2 electron produce H2O, right? What they have written over here in theory is that they have multiplied this whole equation with a 2 so that we can, cancel out this half and this becomes 1. So 4H positive plus oxygen plus 4 electron produces 2 molecule of water. This can also be the equation. We can also write the equation like this. Okay, so now 2 hydrogen are being pumped into the intermembrane space. Complex 3 was pumping how many hydrogen? It pumped 4 hydrogen. You need to remember the amount of hydrogen which are pumped. Okay, complex 1 is pumping 4 hydrogen. Complex 2 is not pumping any hydrogen. It is just transferring electrons. Complex 3 is again pumping 4 hydrogen. Complex 4 is pumping 2 hydrogen. Okay, and ATP synthesis is not pumping any hydrogens. Okay, next we have the proton gradient and the ATP synthase. As electrons flow, hydrogen ions, they are pumped into the intermembrane space and this proton gradient is created. ATP synthase is known as complex 5, right? And it helps to convert ADP into ATP. Why? Because phosphate is being added, right, in order to produce ATP. All right, now what happens to the energy yield? So this is a thing that you need to learn because if you're gonna, understand the whole basics behind it, there's actually a whole molecular mechanism behind how NADH produces 2.5 ATP and how FADH produces 1.5 ATP. That is related to the MBBS level. So you don't need to remember it right now. You just need to remember that NADH, it produces 2.5 ATP and FADH, it produces 1.5 ATP each. Okay. From one glucose, what are produced via glycolysis and Krebs cycle, we have got 10 NADH and 2 FADH that are produced. Okay, if I combine the yield of glycolysis and Krebs cycle, then they are saying that. So if I multiply it by 2.5, it produces 25 and 2 FADH multiplied by 1.5, it produces 3 ATP. At the substrate level, we produced 4 ATP, right? substrate level, I'm talking about the substrate level phosphorylation where succinyl CoA was being converted into succinate, right? So if we multiply it, what happens is that 4 ATP are being produced and the total number of ATP are 30 to 32 ATP. Basically what they ask you what is the energy yield after glycolysis and Krebs cycle. Okay, so what are you going to say? The total energy yield which is produced via these processes, it is 30 to 32 ATP. Okay, wait a second. Where did it go? Yeah. All right, now substrate level phosphorylation. What happens is that it is the direct synthesis of ATP or GDP from ADP. ADP plus phosphate on inorganic plus a high energy substrate. This phosphate combines with ADP in order to produce ATP plus our product. Okay, and this is occurs at the level of the this basically occurs during a metabolic reaction. I'm so sorry, I just lost words. So, substrate level phosphorylation occurs in glycolysis at the level of this. Okay, 1 comma 3 bisphosphoglycerate is converting into 3 phosphoglycerate. Wait, let me just share the diagram with you. Yeah, wait a second. Okay, let me just erase this a bit. Okay, so at this level, they're saying at this level, 1 comma 3 bisphosphoglycerate is converted into 3 phosphoglycerate. This is known as substrate level phosphorylation. Why? Because it yields ATP. Secondly, when phosphoenol pyruvate is converted into pyruvate, it also yields ATP. So this is a substrate level phosphorylation that produces 2 ATP per glucose from glycolysis. Now in the Krebs cycle, where was substrate level phosphorylation? Do you remember? It is right over here. This is known as substrate level phosphorylation where ATP is being produced. Succinyl CoA converts into succinate in order to produce ATP, right? So what happens is that it is converted and 2 ATP are produced per glucose. So 2 from glycolysis plus 2 from the Krebs cycle, it produced total of 4 ATP per per glucose molecule per substrate level phosphorylation. So this is the substrate level ATP which I was talking about, that how 4 ATP are produced. I just explained it to you here. Okay, now we move on to the cellular respiration of fats and proteins. Yeah. So cellular respiration of fats and proteins. What happens over here? We have got lipids. Fats, basically, these are triglycerides and these are made up of glycerol plus fatty acids. Now, what is the glycerol pathway? Glycerol converts into G3P via glycerol kinase. Now, this G3P is produced at the level of glyceraldehyde 3 phosphate, right? And it generates ATP and NADH like glucose. I'm going to show you G3P. See. This is G3P, which is produced after the oxidation of fats and the metabolism of fats, and then it follows this cycle in order to produce ATP over here. So eventually, all of these molecules are being converted into what? All of these molecules are converted into pyruvate. Okay. All right. Now there is also beta oxidation of fatty acids. By beta oxidation, it is broken down into 2 carbon acetyl CoA. So when acetyl CoA is produced, what happens is that it easily enters the Krebs cycle. Then it produces NADH and FADH. Then it goes into electron transport chain. So there are basically 2 pathways of glycerol and fatty acids. The first one, see. Fats are being converted into glycerol and fatty acids. Both of them have different pathways. See, okay, glycerol converts into G3P. It starts from glycolysis and then it goes into the Krebs cycle. Whereas what happens in fatty acid, fatty acids undergoes beta oxidation. After beta oxidation, it produces acetyl-CoA and it directly starts the Krebs cycle. So it does not go through glycolysis. It directly starts the Krebs cycle, and after that it produces electron transport chain which yields ATP, right? Fat yields more energy than carbohydrates. 1 gram of fat releases 9 kilocalories, whereas 4 kilocalories are released for carbohydrate. You need to remember this. This is a very important point for your MCQ point of view, that 1 gram of fat releases 9, whereas for carbohydrate, they release 4 kilocalories. For example, palmitic acid, it produces 106 molecules of ATP. Right. Next, we have proteins. What happens in proteins is that these are converted into amino acids, and then what happens? Deamination occurs. Deamination. D means removal. Amination means the amino group, the classic amino group, which is NH2. So it is the removal of amino group. It occurs in the liver, and this is basically known as the urea cycle, where amino group is being converted into urea, and then it is excreted out. Okay. Next, we have got the carbon skeleton, that is the keto acid. It enters respiration at different points, right? If it forms pyruvate, then it enters glycolysis. If it forms acetyl CoA, then it enters the Krebs cycle. And if it is an intermediate of the Krebs cycle, still it comes and joins in between, and it completes the Krebs cycle. Okay, so if there is a keto acid, is basically does what is that wherever whatever product it forms, for example, it forms pyruvate, it starts from the end of glycolysis. If it forms acetyl CoA, it starts from the whole Krebs cycle. If it is at any point in the middle of the Krebs cycle, it starts directly from there. So there is no fixed rule for this. Okay. And I was basically explaining you this. This is the structure of an amino acid. This is the amine group, right? Which is going into the urea cycle. Okay, and this will be deaminated. NH2 group will be easily removed, and after urea cycle, what happens? This will be removed from the body. Whereas this carboxylic acid group, this group, right, this will undergo keto acid. This can enter as pyruvate, this can enter as acetyl CoA, or this can enter as any intermediate, alpha ketoglutarate, succinyl CoA, fumarate, or oxaloacetate. So we are done with the whole chapter of bioenergetics. Let me know if you guys have any queries. Let me just give you a quick recap. We have done acellular life as well. If you haven't seen that video, please go and check that out. And also we have done bioenergetic respiration, outline cellular respiration of proteins and fats, and correlate these with that of glucose. We just did what happens in proteins and fats, right? Carboxylic acid group, and urea, deamination of urea separately for proteins, and beta oxidation for fats, right? And in glucose, we studied all of the mechanisms: glycolysis, linking reaction, Krebs cycle, electron transport chain, right? Also, we studied about cellular respiration. First, we started from the definition of bioenergetics. We started from cellular respiration, then we did the difference between aerobic and anaerobic respiration, right? Then we move on to anaerobic respiration, and we explained the difference between alcoholic and lactic acid fermentation, right? We did these two diagrams. Then we moved on to aerobic respiration and on the glycolysis, energy investment and yielding phase, and after that this whole chapter we did today. If you haven't seen the first video, it has been uploaded on my YouTube. Please go and check that out so that your concepts may be clear. And if you guys have any queries, please let me know so that we can solve it together. And I'm ending the video now, and all of the lectures of MDCAT have been prepared in accordance with the 2025 MDCAT syllabus, and your books have been followed. The local system books have been followed, which are Federal Board, Sindh Board, Punjab Board, and KPK Board, right? So still, if you guys think that any suggestions, any queries, anything, please let me know. Until then, take care and bye-bye.